Hello everyone, and welcome to another recorded lecture of BIO101 online. Today we will talk about Chapter 10, which is Meiosis and Sexual Reproduction. We spoke about reproduction in the first class, actually. We said that reproduction can be either asexual or sexual. And in asexual reproduction, we see that one parent organism replicates its DNA and simply splits into two cells. So this should remind you of mitosis, right? One parent organism copies its DNA and then splits into two cells. And when you do this asexually, we get two genetically identical offspring. So for example, here's an amoeba parent. This parent has to double its DNA. They split. And that's it. That's the end. Now we have two daughter cells that are identical to the original parent. So this is asexual reproduction. It's the same idea as mitosis or binary fission. But reproduction can be sexual as well. And in sexual reproduction, the DNA of the offspring comes from two parents, not just one parent. And what we'll see in sexual reproduction is that an egg from the mother fuses with a sperm from the father, which gives us the first cell of the next generation. And in sexual reproduction, we can generate genetically diverse offspring. Specifically, there are more than 70 trillion different genetic combinations possible when two individuals mate. But why do we care about sex? Why do we care about sexual reproduction if asexual reproduction costs a lot less energy? Right? Attracting a mate and producing sex cells requires a lot of energy input, so it's costly overall to the organism. So take a second to pause here and think about why sexual reproduction is so common and so beneficial. Sexual reproduction produces genetic diversity among offspring. Right? Whereas in asexual reproduction, we got a basic clone of the parent. With sexual reproduction, we get offspring that are genetically diverse from either parent. And diversity will increase the chance that some offspring will survive in a changing environment. So genetic diversity is worth the cost of sex for many organisms. So I'm going to give you an example. Suppose you have a population of organisms that are not very diverse. You have a genetically identical population. And over time, let's say there's this disaster. There's some type of climate change or some kind of earthquake. All of the organisms will die. That's the end of that organism. However, in a genetically diverse population where you have different gene combinations to start, right? So you do have, of course, these main offshoots of populations that are similar to each other. But when you do have sexual reproduction, you're mixing and matching different genes. So suppose there is an ecological disaster, most of the population might be destroyed. But because there are some diverse combinations of genes, those little offshoots might be able to reproduce into new populations. And over time, right, with sexual reproduction, we can survive in ever-changing conditions. So this is why sexual reproduction is worth the effort. So what are you made of? You're made of two different types of cells, and we spoke about this before. Somatic cells are body cells. Those are diploid, meaning they have two copies of every gene. Somatic cells are your skin cells, your hair cells, your eye cells, your muscle cells, blood cells. All those are your somatic cells. And all those cells have two copies of each gene. So you have a hemoglobin gene. That means every one of your hair cells has two copies of a hemoglobin gene. All right? The other type of cells you have in you are gametes. And gametes are sex cells, sperm or egg. Gametes, unlike somatic cells, have half the number of chromosomes, right? So somatic cells are diploid, they have two copies of each gene. Gametes are haploid, they only have one copy of each gene. So a sperm or egg is only going to have one copy of hemoglobin, for example. Gametes are not located all throughout the body. They're only located in the gonads. So in females, that means ovaries. In males, that means the testes. And the gametes are what can pass on DNA to the next generation. I actually just want to take a second to make sure you understand why is it important that sperm and egg are both haploid, right? What would happen if sperm and egg were diploid? Right? If they both came together and they were both regular diploid cells, then the new generation would be tetraploid, right? It would have 4 copies of every gene. We don't want that. That's a disaster. So we want to make sure that sperm only have 1 copy, egg only have 1 copy. So when they come together, we can have the nice diploid start to the new organism, right? We want the organism to have 2 copies of each gene. So our genome, which is contained in every cell in our body, right, with some exceptions, but our genome, so all of those genes, all the chromosomes that we have, are found amongst 23 pairs of chromosomes. So the genome is the entire collection of genes, right? All of our genes are found in 23 pairs of chromosomes. We spoke about chromosomes last time, that they're DNA molecules wrapped around proteins. We have 23 pairs. 23 of these came from mom, 23 of them came from dad, but they all have pairs. So we're going to talk a lot about what that means in this lecture. So there are 23 pairs of chromosomes, 46 in all. Right, we have 46 chromosomes in all. And chromosome pairs 1 through 22 do not influence sex at all. We call those chromosomes autosomes. So chromosome pairs 1 through 22, or chromosomes 1 through 44, are all autosomes. In mammals, the sex chromosomes X and Y determine sex. So the 23rd pair of chromosomes in humans is the sex chromosomes. And if you have 2 copies of the X chromosome, that would make you female. And if you have a Y chromosome, the presence of a Y chromosome in humans would denote male in biology. So the entire genome, whether you're in a hair cell or a muscle cell, has 23 pairs of chromosomes, right? And those 23 pairs of chromosomes have all of the genes that make you. So all of your genes that make you are just divided up into these 23 pairs of genes. So altogether, since there are 23 pairs, that means there are 46 chromosomes, individual chromosomes. You can count there are 46. So half of these came from mom, and half of them came from dad. And when all the chromosomes of the genome are laid out like this, and they're all stained with a special dye, that's called the karyotype. So this is a karyotype of an individual. It is all of the chromosomes in the genome laid out. That's a karyotype. So this karyotype is taken from A, male, female, or impossible to determine. You can pause here. And the answer is male, because of the presence of the Y chromosome. This karyotype shown is taken from A. Is it a gamete in G1, a somatic cell in G1, a gamete in G2, or a somatic cell in G2? So you can again pause here. And remember that in between G1 and G2 is S phase, where you get double the genetic information. And looking here, since we see 23 pairs of chromosomes, including an X and a Y, we know that this is a single set of DNA. Right? This has to be a single set of DNA. So it has to be a cell that's in G1. This is not a cell that's ready to divide. So it's not going to have to go through S phase and be in G2. Right, so this is definitely in G1. And since we have 46 chromosomes, 23 pairs, we know this is a diploid organism. So the answer has to be a somatic cell in G1. Once this somatic cell goes into G2, then of course each will have a separate copy. So that will copy like that will copy, and then again this will copy, that will copy. So we're going to end up with 92 individual chromatids, right? Each of these individual pieces is a chromatid. But we could also call those a chromosome. I want to take a moment to talk about something that's not in the textbook. But there's a common misconception that genes determine gender, and gender is very different than sex. So sex refers to the biological and physiological characteristics that define men and women. So key is physiological and biological. So these are determined by chromosomes. This is where sex chromosomes come in. There are factors on the Y chromosome that determine the production of testosterone. So the presence of different hormones will greatly influence the structures of genitalia. And if you have testes in biology, and you produce sperm, we denote that the male. The organism that produces the egg is usually the female, right? And the way that we can simplify this is that in all organisms, right? Because plants, for example, make pollen. We don't call the male gamete a sperm cell if it's a plant. We call it pollen. But in all of biology, the sex that makes the larger of the two gametes is called the female. So that's sex. So since the egg is so much larger than the sperm, right? We know that the egg-producing individuals are called females, right? So and since pollen is a lot smaller than ovules of a plant, right? The parts that make the sperm, the smaller of the two the smaller of the two gametes are going to be called the male parts. The parts that contain the larger gamete will be the female part. So sex is a biological and physiological thing. Gender is not. Gender is more of a societal construct, right? So gender refers to socially constructed roles and behaviors and attributes that a certain society and a given place and time deems appropriate for one sex. So gender is influenced by chromosomes and hormones, but everyone's influenced differently. So gender is not determined by biology per se. It has a lot to do with the environment as well. And this is not on the quiz, but there's a really good book called Evolution's Rainbow by Joan Ruff-Garden that talks about the diversity of different genders in the animal world, and talks a lot about the evolution of sexuality. So there's a couple of myths that I want to debunk. So true or false, an organism is solely male or female for life. So this is a false statement. Many plants and many animals are both male and female in the same lifetime. Males are bigger than female on average. That is also false. In many species, especially in fish, the females are always larger. Females give birth and not males. That's false, right? And some species, the females deposit their eggs in the pouch of a male, and the male actually gives birth. Males always have an XY chromosome, and females have XX. Is this true or false? This is false. It's actually the opposite in birds. They don't have XY, they have ZW. And some species don't even have sex chromosomes. And sex is determined by temperature, for example, in like alligators and other reptiles. Only two sexes occur in nature. That is very false, right? Some can have 3 or more sexes or can be intersex. Males have penises and females lactate. That's also false. For example, female spotted hyenas have a penis, and fruit bats actually, male fruit bats lactate. And males are dominant and control females. This is also very false. And in many species, females control males and avoid dominant males, and dominant males are selected against. So here's just a little bit of perspective on the diversity of the animal and plant world. Now let's give a brief refresher on chromosomes. Chromosomes, we said, are made of chromatin, right? Which chromatin is DNA wrapped around histone proteins. So here we have a DNA double helix that's made up of nucleotides, right? Those nucleotides are linked together in a chain, and then two of these chains form a double helix, right? They're connected by hydrogen bonds. This whole double helix gets wrapped around histone proteins many, many times. This is one chromatid. This would, after replication, it would look like this. And these individual things would be sister chromatids. These would be identical to each other and attached at the centromere. So a quick refresher, you can pause here and answer this question. So the answer is B, histones. And you can pause here. The answer is B, centromere. Okay, you can pause here for this question. The answer is C, S phase. Right? S phase has to happen to duplicate all of the chromosomes. Chromosomes would appear as in picture 5 during blink of the cell cycle. So the answer is only D, during prophase. Right? Condensed. Okay. So now I want to talk about homologous chromosomes. Like I said, humans have 2 copies of each chromosome, 1 through 23, giving us 46 in total. 1 copy of each chromosome came from mom, the other came from dad. So for example, you have 2 copies of chromosome 15 in your somatic cells. One of those copies came from your mother, the other copy came from your father. Because both copies of chromosome 15 are essentially the same in terms of size and the order of genes, we say that those are homologous. Right? Homologous means the same in relation to each other. So, for example, if this is this is an example, right? Not real. Suppose these are 2 copies of chromosome 15. Chromosome 15 from mom is here. Chromosome 15 from dad is here. Suppose that, right, so we call these homologous because suppose that there are 3 different genes. One is for eye color, one is for enzyme A, and one is for an enzyme called cytochrome C. Right? These genes are all found on chromosome 15. So the chromosome 15 you got from dad and the chromosome 15 you got from mom will have the same genes in the same locations. So now, does this mean that they are identical? So is chromosome 15 from mom identical to chromosome 15 from dad? So no, right? They're not identical. They just have the same types of genes in the same locations, but this might be a different form of a gene than this form. So one single gene might have different forms or different flavors. We call these alleles. So an allele is a version of a gene. So, for example, the gene for eye color can have a blue allele for blue eye color, or can have a green allele for green eye color. Right? It's still the eye color gene, but it could be different flavors of eye color, right? You could have black eyes, you could have brown eyes, you could have hazel eyes, etc. So each of those colors would be different alleles. For enzyme A, suppose you have a healthy copy, that's one allele, one version, or you can have a mutant enzyme A. right? That's a different mutant allele, a different version. So what I'm trying to illustrate is that homologous chromosomes have the same genes in the same location, but the alleles might be different, right? Dad might have a green-eyed allele, but on his chromosome 15, but mom might have a blue-eyed allele on her chromosome 15 in the same location. These are homologous chromosomes, right? So what happens to these chromosomes in S phase of interphase? Chromosomes are duplicated by DNA polymerase, resulting in two identical copies. So chromosome 15 from dad was duplicated, chromosome 15 from mom was duplicated, and what do we call each of these copies now? Each copy is now called a sister chromatid. And sister chromatids are identical. So this sister chromatid is identical to this chromatid, but this sister chromatid is not equal to this one. So I would call this and this non-sister chromatids, but they are homologous because they have the same genes in the same location. I'm going to go over this on the next slide, but this is something that gets a lot of students confused. Homologous chromosomes versus sister chromatids. It is the most important thing to understand in this chapter. So again, I'm going to show you a different picture that maybe helps you in a different way. So during S phase of the cell cycle, we said chromosomes are replicated by DNA polymerase, and a duplicated chromosome consists of two identical sister chromatids. So this from mom has been already replicated, right? This from dad has been already replicated. So this pair and this pair are homologous to each other. But within each pair, right, within each duplicated chromosome, there are two identical sister chromatids. So this is identical to this. This is identical to this. But this is not necessarily identical to that, right? So this is homologous with this. And genes are always located on specific positions. So in homologous chromosomes, like this gene is the same as this gene. This gene is the same as this gene. Of course, there are different alleles, possibly, but they're all ordered in a very specific location. And we have now gene maps. So gene maps are the results of lots of genetic studies that show the relative positions of genes on a chromosome. So Drosophila only have 4 chromosomes, and on chromosome 2, there are a lot of different genes. For example, at this location, encodes for either light-eyed or purple-eyed. At this location, it encodes for either a small wing or a curved wing. Right. So over here, it encodes for bristles or not on the antenna. So we know exactly where certain genes are located in different organisms. So for example, the map of human chromosome X here shows a lot of different genetic diseases that are X-linked, that are linked to the X chromosome. So take a look at this rapid response question and tell me which statement is true. So pause here. So we'll go one by one. Both came from the person's mother. No, that's not correct. The 1-2 pair is identical to the 3-4 pair. No, that is not true. 1 is identical to 2. That is correct, right? So C is correct. The chromosomes have been replicated already. Right, so we can say that these are homologous to these, and 3 and 4 are sister chromatids, and 1 and 2 are sister chromatids. But 2 and 3 are non-sister homologous chromosomes. How many copies of chromosome 10 do you have in your cells during G1 of interphase? I'm assuming that these are somatic cells. So how many copies do you have during G1 phase of just one chromosome 10? Right? If it's a somatic cell, we have 2 copies of chromosome 10. So what is the difference between the two copies of chromosome 10 and interphase? Right, so the answer is B, right? One copy of chromosome 10 came from mom, the other copy of chromosome 10 came from dad. How many copies of chromosome 10 do you have in your somatic cells in G2 phase? Right, so remember, DNA gets copied in S phase, which is between G1 and G2. So if we had two copies of chromosome 10, right, one from mom and one from dad, in G1, that means in S phase, those two copies got duplicated, which means we would have C, 4 copies of chromosome 10 after the DNA is copied. So let's just review haploid and diploid cells. So how many chromosomes do human skin cells have? Right, we have 46 total, 23 pairs of homologous chromosomes, right? We said one copy of each chromosome came from each parent. How many chromosomes do human sperm cells have? 23 single chromosomes. They only have one set, and that makes them haploid, right? They only have one copy of each chromosome. So this brings us to meiosis, which is part of the human life cycle. So when a haploid egg is fertilized, here's my egg and here's my sperm. When a haploid egg and a haploid sperm fertilize each other, we get the first cell. The fertilized egg is called the zygote. That's an important word. So the fertilized egg is called the zygote, and it's the first diploid cell of the new generation. Right, so two haploid genomes come together to form a diploid genome. This diploid genome undergoes mitosis and embryonic development to make multiple copies of cells with the same DNA. Once in puberty, the adult will then undergo meiosis once again. Meiosis produces gametes that are haploid that can then recombine to form a new diploid organism and start the next generation. So mitosis is responsible for growth, repair, and development. And chromosome number must be maintained in somatic cells, right? If you want to make more functional cells, you don't want to get fewer and fewer chromosomes. You want to make sure all the cells have identical genetic information. So in mitosis, we're producing genetically identical cells that are all diploid. And mitosis occurs throughout the body and throughout the lifespan of an organism. So we do mitosis since we're a zygote. Meiosis is a totally different story. So meiosis divides chromosomes in half. And you could say meiosis or meiosis. I'm going to go interchangeable. So in meiosis, we get haploid gametes made from special germ cells that are called, they're called germ cells, but they're special sex stem cells. And what makes them special is that these stem cells can only produce gametes. So all gametes are formed from special diploid germ cells. In male animals, we call them spermatocytes. And in human females, we call those oocytes. We'll talk more about that in a bit. But what I want you to understand now is that meiosis is going to help us divide chromosomes in half so we can form haploid gametes, right? Where mitosis gives us equal cells that all have 46 chromosomes. Meiosis does not do that. Meiosis gives us half the number. So it's called reductional division. This does not occur throughout the body. It only occurs in the gonads, right? And these haploid cells are all genetically unique from each other. And we'll talk about how they acquire this genetic uniqueness, right? Because this is what's going to allow for all the diversity in sexual reproduction. Right? If you have a lot of diverse sperm and a lot of diverse eggs, you can have a lot of diverse zygotes. And unlike mitosis, meiosis can only be completed after puberty. So whereas we do mitosis from the day we're conceived to the day we die, meiosis is only done after puberty. And in females, meiosis stops during menopause. So suppose a zygote of a ferret contains 40 chromosomes. How many chromosomes would be in a ferret's sperm cell? So you can pause here. And the answer is B, 20. Right, same would be in a female ferret's egg cells. What is the chromosome number of a human sperm cell not in meiosis? So a normal human sperm cell would have 23 single chromosomes. So meiosis consists of 2 cell divisions. And remember that only special diploid germ cells in the gonads are going to do meiosis. So these special cells, whether it's in the ovaries or the testes, go through 2 cell divisions to make haploid gametes. We call these meiosis I and meiosis II. Meiosis consists of interphase, followed by meiosis I. Again, we have the same phases as in mitosis. We have prophase I, metaphase I, anaphase I, telophase I. Then there is a cytokinesis, referred to as interkinesis in the textbook. We then have meiosis II, which is prophase II, metaphase II, anaphase II, and telophase II. And then another cytokinesis. There is no second interphase. So there's no replication of DNA in between cytokinesis and prophase II. Right? So in between here, there's no copying of the genetic material, which is going to allow us to divide it in half. Right? So if we start with 1 copy of each gene, right? 1 from mom, 1 from dad, those are each going to duplicate. And at the end, we're going to end off with a single copy in 4 separate cells. So we're going to get 4 haploid cells as a result of 1 cell going through meiosis. So 1 diploid germ cell goes through meiosis, and that's meiosis I and meiosis II, and we're going to get 4 genetically diverse haploid gametes. Right, so that's key. I want to make sure that's very critical. So we're going to get 4 haploid gametes from 1 special germ cell. So meiosis I and meiosis II each have 4 phases that are very similar to mitosis, but there are some important differences, especially in meiosis I. I'm going to give you a brief overview before we dive into the details. In interphase, all chromosomes are replicated. So each of the 46 chromosomes will consist of 2 sister chromatids. So we have 92 chromatids in the cell. We now have a special thing happening in meiosis I. Each of the homologous pairs will meet with each other. So you have a duplicated chromosome 1 from mom, a duplicated chromosome 1 from dad. Those are going to meet each other and exchange genes. We're going to talk a lot about that in a bit. And then they're going to go their own way into 2 separate cells. Now, each cell is only going to have 1 version of each gene. So these are no longer diploid. After meiosis I, we have haploid cells. And each haploid cell still has a duplicated chromosome. So in meiosis, what we're going to do is just separate those sister chromatids. And at the end, we will have 4 haploid cells with unduplicated chromosomes that are different from each other. They're all a little bit different from each other. This is, of course, a little confusing because there's only one chromosome here. We have 46, I'm sorry, we'll have, 46 of these in each of our cells. So how is genetic diversity achieved in meiosis? We keep on talking about how important sex was to get genetic diversity, but how is that actually achieved? So during crossing over and recombination, two duplicated homologous chromosomes pair up and they physically exchange pieces, which can scramble the genetic information. So here, let's zoom in to a special germ cell of this frog. So there's a special, it will go into the testes of this frog. And here is a diploid cell that's meant to make sperm. So it's meant to make sperm for the frog. And in this diploid cell, it already copied its genetic information in interphase. So now it's in prophase I of meiosis. So let's zoom in on one of these pairs of chromosomes in the frog. So here we have a homologous pair of chromosomes. Remember, this is duplicated already. Normally, you would only have one copy of let's say this is chromosome 1 from dad, and one copy of chromosome 1 from mom. But since it went through interphase already, right, each chromosome has a sister chromatid that is identical, right? And on this one chromosome, there are 6 different genes, but mom and dad have completely different alleles for each of the 6 genes. So there's genes A, B, C, D, E, and F, but mom has the lowercase alleles, dad has all the uppercase alleles. So what I'm going to try to show you is that in meiosis I, there's crossing over, which can combine the different alleles, allowing the different gametes to have different combinations of genes. So what's happening over here is there's crossing over, there's physical exchange of genetic information. So here you can see that genes A and B are being exchanged. And this is in meiosis I. And after meiosis I, we now have haploid cells, right? These are now haploid. We only have one copy of each gene. We don't have 2 copies anymore, right? Before, again, with the exception of this crossing over, before we had big D and little d, right? Now we only have big D or little d, right? So after meiosis I, we have 2 cells that are no longer diploid. You either have one copy or the other, with the exception of this additional genetic material from crossing over. In meiosis II, these sisters will separate, forming 4 haploid gametes. Some of the gametes will have the same sets of genes as one of the parents. So for example, this is a parental gamete, meaning it's the same as one of the parents. It has all capital letters like dad had, right? This one had all lowercase letters like mom had. But with recombination, right, from crossing over, you can have recombinant gametes that have different combinations of alleles from mom and dad. So this is, again, what gives us the first level of genetic diversity. We're mixing and matching genes. And this all happens in prophase I, right, of meiosis. This does not happen in mitosis. So these are 2 homologous replicated chromosomes. We call, when they line up in prophase I, they swap genes, right? But first they have to form a tetrad. This is called a tetrad, when the 4 chromatids are lined up. Tetra means 4. It's also called the bivalent in the book, right? So the duplicated homologs line up and form a tetrad, and they do crossing over. They do like a little handshake and exchange genetic information. And these 2 in the middle would then be recombinant chromatids, right? Because they're unlike either of the parents. So this is what we went through so far. In interphase, DNA replicates. The cell produced all the proteins needed for cell division. Then we went into prophase. And in prophase, chromosomes condense, the nucleus starts to break down, the mitotic spindle forms, so same thing as in prophase of mitosis. But in meiosis I, we have crossing over occurring. That's the biggest difference in prophase of meiosis versus mitosis. So this is when homologous chromosomes physically exchange genetic information, right? And in metaphase I, all of the paired homologous chromosomes line up along the equator of the cell. This is very unique. Remember, in mitosis, the microtubules grabbed on to each sister chromatid separately and pulled the sister chromatids apart. But in meiosis, in metaphase I of meiosis, we have the homologous chromosomes lining up, and each microtubule only grabs onto one pair. So we're going to zoom into this up close in a bit. But each microtubule is grabbing onto one pair of homologous chromosomes. And in Anaphase I, we're going to see homologous chromosomes separate. So let's look closely at what we're seeing in metaphase I. But remember, it's homologous chromosomes are lining up in 2 rows. So that means chromosome 1 from dad and chromosome 1 from mom is here, right? Chromosome 2 from dad, chromosome 2 from mom is here. They're all lined up with their partner after they exchange genetic information. In metaphase I, there's a phenomenon called independent assortment, which again gives us a lot of genetic diversity in the gametes. So the way that the mom and dad's chromosomes line up is completely random. So yes, you can have all of dads on the right, on the top, all moms on the bottom, and then you can have, after meiosis, the gametes have all of dad's alleles and all of mom's alleles. And this is, of course, assuming that no crossing over occurred. So this is without even taking crossing over into consideration. You can also have 3 other alternatives, right? Depending on how mom and dad's chromosomes align at the metaphase plate. So you could have any of these alternatives, which would give you different combinations of chromatids in the gametes. So in a germ cell that has 3 pairs of chromosomes, there are 4 possible arrangements during metaphase, right? It could arrange this way, all on top versus all on the bottom. You could have the 3rd pair mismatched, right? You can have the 2nd pair mismatched or the 1st pair mismatched. So there's 4 different arrangements at metaphase. How many possible chromosome arrangements are possible in metaphase I of humans? That would be 2 to the 23rd power. So chromosomes have over 8 million different ways to combine. And this is without taking crossing over into consideration. So I hope now you're starting to understand how we're achieving this genetic diversity in meiosis as we produce these sperm or egg. So take a look at this video now. This is found in the Connect website.
Crossing over during meiosis allows for recombination of genes between homologous chromosomes. This alters the arrangement of genes on the same chromosome. Crossing over occurs frequently during prophase I of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent, and the other member came from the other parent. In this example, one parental chromosome had alleles uppercase A and uppercase B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of genes is not altered in this case. Now consider the case where there is crossing over in the region between the two genes. Since the crossing over is only between two individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over, and will still have the parental combination of alleles, either uppercase A and uppercase B, or lowercase a and lowercase b. The other 2 will be recombinant, with either uppercase A and lowercase b, or lowercase a and uppercase b alleles. Thus, crossing over shuffles the alleles and brings about genetic recombination in the gametes.
So during fertilization, we have chromosomes donated by 2 different parents. If we take 2 different parents into account, that gives us 70 trillion chromosomally different zygotes possible. Right? We said each individual human could produce about 8 million different gametes. So when we get 2 of those in fertilization, right, the union of male and female gametes gives us more than 70 trillion. That's all assuming that there's no crossing over. So if there's 1 crossing over event, that would give us 4 to the 23rd power squared. Or this number over here, different amounts of zygotes available. So the fun fact is crossing over may occur several times at each chromosome. So you can imagine how many genetically different zygotes are possible. And this is why all of us are truly unique. I wanted to take a second to just think about what these letters really mean. Suppose there are 3 genetic diseases encoded by genes A, B, and C. There's a healthy version and a non-healthy version of each. So in the case of gene A, having a big A, capital A is healthy. Having a little a is the disease. Big B is healthy, little b is the disease. With C, little c is actually healthy, but big C is the disease. And we'll talk a little bit more about this in genetics, how different versions of a gene might denote the healthy version or the unhealthy mutant version. So if there were no crossing over, right, that means, let's say this came from mom and this came from dad. Dad has disease C only. Mom has diseases A and B. If there were no crossing over, that means all the children, all the gametes would always be diseased. With crossing over, so this is during interphase, we have the 2 homologs duplicate. During prophase I, we have crossing over we spoke about. Right? And then after meiosis, we can actually have a possibility of a gamete with no disease at all. Right? You have big A, big B, and little c. Because there is a crossing over right over here, right? This exchange of genes gave us different possibilities. So this sperm or egg would actually give rise to a completely healthy individual. This is a completely hypothetical example, but you can imagine how mixing and matching of genes gives us more possibilities, which means more chances of survival in a changing environment. So we've made it all the way to anaphase so far, right? So make sure you look at this very closely. Make sure you go through this. Of course, you've done the reading already, so this is not brand new information. But we said that in prophase, the chromosomes condense to become visible, right? The crossing over occurs, spindle starts to form, the nuclear envelope breaks up, the nucleolus totally disappears. During Metaphase I, the bivalents, the tetrads, are held together by their chiasmata. Their chiasmata are the locations that they cross over. They're called chiasmata. Those little X's where they cross over. And at this time, we have microtubules attached to each homologous chromosome pair. And we said that the bivalents independently align themselves at the metaphase plate. So either the maternal or paternal homolog of each bivalent could be oriented toward either pole of the cell. So that's called independent assortment. And that's what gives us lots of genetic variability in the daughter cells. So in Anaphase I, the homologous chromosomes separate to separate poles of the cell, but the sister chromatids remain intact. So that's key. In Anaphase I, homologous chromosomes separate. Underline homologous chromosomes. Because in Anaphase II, we're going to see the sister chromatids separate. So in Anaphase I, homologous chromosomes separate. In Telophase I, the chromatids still remain together, but they temporarily decondense. Two nuclear envelopes form around the chromosomes, and the spindle starts to disappear. So telophase is like the reverse of prophase. And there's cytokinesis, which divides the cell into two. So let's watch this video that will take us to Meiosis II, which is where we left off.
During the S phase of interphase, the DNA is replicated, producing two copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters Prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but two events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during Prophase I, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase I concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers forming the meiotic spindle. In Prometaphase I, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules.
So this is really critical. This is what I want you to understand. One microtubule from one side is pulling onto this blue pair. Another microtubule is holding onto this purple pair. Right? So they're pulling pairs of chromosomes apart during anaphase. That's different from anaphase and mitosis, where we're just separating sister chromatids, right? And each centromere had a separate microtubule attached to the kinetochore. Here, only one kinetochore of each pair of homologous chromosomes is attached, right, to the mitotic spindle.
In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During Metaphase I, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In Anaphase I, the homologous chromosomes separate and move toward opposite poles. Meiosis I ends with Telophase I, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow.
So once Meiosis I is complete, we are ready to go on to Meiosis II. And Meiosis II has the same four phases, right? But we call them Prophase II, Metaphase II, Anaphase II, Telophase II. And at the end of Meiosis I, we finish with two daughter cells that have the haploid number of chromosomes, meaning they only have one chromosome from each homologous pair. In this fake organism in this picture, N equals 2, meaning there are only 2 chromosomes in this genome. For humans, it would be 23, right? N equals 23. So at the end of meiosis 1, you would have 23 of these chromosomes, but you only have one, right? One of each pair, either the one from mom or the one from dad. Of course, it's possible that some of the pieces mixed and matched from mom and dad during crossing over. So it's possible that at the end of meiosis 1, you might have a cell with a chromosome with mom and dad's genes. Like that's shown here in this picture. It looks like crossing over has occurred here in one of these chromosomes. But otherwise, we have 2 haploid cells. But what's interesting about these haploid cells is that they have 2 sister chromatids. So each of these sister chromatids are copies of each other, and we have no need for those copies, right? We only want one copy of each gene in our haploid gametes at the end of meiosis 2. So what we're going to do is we're going to pull apart these sister chromatids so we end up with haploid cells. So in Prophase II, same idea. The spindles are going to form from the centrosome, right? They're going to start moving to opposite poles of the cell so they can grab onto those chromosomes, right? The nuclear envelope dissolves, the chromosomes condense. And in Metaphase II, the chromosomes line up along the midplate of the cell, very much like they do in mitosis, right? In meiosis 1, it was very different kind of metaphase, right? In Metaphase I, homologous chromosomes lined up, and the pairs together were pulled to either section. But in mitosis, we saw that each kinetochore was attached to part of the mitotic spindle, just like we see here in meiosis 2, right? As part of the mitotic spindle, those microtubules are grabbing on to each half of this replicated chromosome. So it's going to pull apart the sisters. So this looks more like mitosis, right? Meiosis 2 looks more like mitosis, right? Meiosis 1 is a different situation. So like I said, in Anaphase II, sister chromatids separate to the opposite poles of the cell, right? They move toward their opposite poles by the microtubules pulling on them, just like in mitosis. So test yourself right now. What was separated in Anaphase I? Right? The answer is homologous chromosomes, right? Homologous chromosomes were separated in Anaphase I, but sister chromatids are separated in Anaphase II. And you can definitely expect that to come up on a quiz or a homework assignment. Next, and finally, we have Telophase II, which is kind of the opposite of prophase. So the nuclear envelope will reform, the chromosomes will start to decondense, the mitotic spindle will disappear, and then in cytokinesis, we're going to end up with 4 non-identical haploid daughter cells. Right? If N equals 2, that means we want to have 2 chromosomes in each. These are haploid, right? Remember that somatic cells have 2 N. So they're going to end up having 4 once a sperm fertilizes an egg. So overall, what we've seen was that meiosis is a special way to mix and match and divide genetic material, right? We're mixing and matching up genes, and then we're dividing them up into haploid cells that can then be recombined to form a new organism. Right? So it's pretty cool how we're doing this. So this video is contained in the ebook. This is a quick one. I'm going to watch this with you. You should watch this over and over again as long as you want to make sure all this content is solidified in your memory.
In Prophase I, the DNA condenses and the homologous chromosomes, consisting of 2 sister chromatids, become closely associated in synapsis. Crossing over between non-sister chromatids may occur during this phase. In addition, the spindle begins to form, and the centrosomes migrate to opposite poles. By Metaphase I, the nuclear membrane has disappeared and the spindle is fully formed. Spindle fibers attach to only one side of each centromere, so that the two homologous chromosomes are attached to spindle fibers from opposite poles. Each pair of homologs then lines up on the metaphase plate. Either the maternal or the paternal homolog may orient toward a given pole. In Anaphase I, the spindle fibers shorten and pull the homologous chromosomes to opposite poles. The sister chromatids do not separate. Each chromosome still has two chromatids. At each pole, there is a complete haploid set of chromosomes, consisting of one member of the homologous pair. During telophase I, the spindle disappears and nuclear membranes reform around the daughter nuclei. Each daughter nucleus contains two sister chromatids attached to a common centromere to form one chromosome. If crossing over has occurred, the two sister chromatids are not identical. During Prophase II, the nuclear envelope breaks down and a new spindle forms. In Metaphase II, spindle fibers bind both sides of the centromeres. During Anaphase II, the spindle fibers contract and the sister chromatids separate and move toward opposite poles. In Telophase II, nuclear envelopes reform, the spindle disappears, and cytokinesis takes place.
Great. So this is an overview of all the stages of meiosis as instructed in the book, and this is the right amount of information to study from. Right, so this is a good phrases to put on flashcards if you're studying that way, or in a Quizlet. So make sure you know what's going on in each stage of meiosis, and you should be able to look at a picture and know what's going on. Right, and here is another way to look at it. I'll go through this with you. We start an interphase with a special sex stem cell, right, called a germ cell. If it's in a male, it's called a spermatogonium. We'll talk about that in a bit. If it's a female, it's called an oogonium. And in the beginning of meiosis, we have the only time DNA is copied. It's only happening once before Prophase I. And in Prophase I, we had that phenomenon called crossing over. So of course, in Prophase I, the nucleus is going to deform, the spindle will start to form, the centrosomes will move to opposite sides of the cell, all that will happen. But something unique called crossing over happens in Prophase I, where the homologous chromosomes physically cross over, and they do what's called homologous recombination. They recombine their genes. And the homologous pairs of chromosomes line up at the equator in Metaphase I, and the meiotic spindle pulls them apart. So the homologous pairs are now separated into two daughter cells that are haploid. They're haploid because they either have one allele of a gene or another. The exception is with crossing over. If crossing over occurred, it's possible that they're non-identical. But we still call them haploid at this point. Because for the most part, they either have most of mom's one chromosome or most of dad's one chromosome. And remember, the orientation of mom and dad's chromosomes in Metaphase I is completely random. So it can be that mom's chromosomes 1, 2, and 3 were on top, but 4 through 23 were on the bottom. And then when you divide these up, you're going to get different combinations every time. It's all random. So between crossing over in Prophase I and independent assortment in Metaphase I, you're getting a lot of diversity already. You divide those two daughter cells, and then they can start Prophase II. There's no DNA duplication, because now we're just dividing these two equally, kind of like in mitosis. So the chromosomes line up at the equator. This time, each half of the chromosome is attached to the mitotic spindle. Right, so the kinetochore proteins are hooked onto the microtubules, and the meiotic spindle pulls the sister chromatids apart in Anaphase II. And then, of course, in telophase, we have the reformation of the nucleus. The chromosomes will unravel, and we're going to have 4 haploid cells that can now become gametes. These are haploid cells that can now become gametes. So meiosis is very different from mitosis. And that's what you should focus on most, I think, more than anything, is focus on how meiosis is similar and different from mitosis. So in meiosis, there's 2 cell divisions. In mitosis, there was only 1. In mitosis, homologous chromosomes never pair up, right? So chromosome 1 from mom and chromosome 1 from dad don't meet up in mitosis. They line up on top of each other, possibly, right, as they're going to be divided. They don't go side by side. They all line up in the middle of the metaphase plate. And of course, in meiosis, we're getting haploid cells at the end. In mitosis, we're getting diploid cells. So these are good summary pictures of the differences. Right. And the book has 3 different tables. One that compares meiosis 1 to mitosis. And you could go through each phase and the differences. So make sure you understand this. This is meiosis 2 compared to mitosis. This is very similar. You're going to notice these are almost the same. But it's good there are a couple of differences, right? For example, at the end of meiosis 2, we're getting 4 haploid non-identical cells. Whereas at the end of mitosis telophase, we're getting 2 diploid identical cells. So these are really helpful. Make sure you understand the differences between the 2 types of cell divisions. And here's a video you can watch on your own that goes through a play-by-play of the differences. I want you to notice, again, I keep on drawing your attention to this. Notice how we're separating the homologous chromosomes and how only 1/2 of the centromere is attached. That's in meiosis 1 only. Now let's compare that to mitosis, where both halves, this half and this half, are both attached to the microtubules, so they both get pulled in half. And again, please watch these videos over and over again at your own pace so you understand this. Right? So in meiosis, we have crossing over and synapsis. Doesn't happen in mitosis. In Metaphase I, the homologs align independently, right? In metaphase of mitosis, they just go one on top of each other, like that. So you would have 46 of these in humans. In anaphase, the homologs separate because it's meiosis. We have homologous chromosomes pair up, so they separate in anaphase I, but in anaphase of mitosis, sister chromatids separate. Right? And then pretty much, telophase is the same idea. But of course, you would see 2 sister chromatids still attached at the end of meiosis 1. Right, so in summary, meiosis goes through 2 rounds of division. The first round separates homologous chromosomes. The second round separates sister chromatids. But the similarities are that they both have the same 4 steps, right? Like maybe 5 if you consider prometaphase. They both use spindle fibers to sort chromosomes. They both use cytokinesis with the help of microfilaments to form a cleavage furrow and pinch the 2 cells apart. So there are similarities. One thing to remember is that there's no DNA replication between meiosis 1 and meiosis 2. Right? There's only one interphase of DNA replication, and that happens before meiosis 1. Right? And the whole point of meiosis is so we get diverse haploid gametes. This is another video that's about 4 minutes long that I won't go through with you, but again, it's in the ebook, and I highly recommend you watch this at least a few times. So now we'll move on to the second half. This is starting from section 10.5 in the Mater textbook. So at the end of meiosis, in plants, what we're going to produce is spores. So they don't have sperm and egg. And this is not only plants, but fungi and some protists, right, produce spores. And these spores are haploid. And there are two different generations to plants. So plants do what's called alternation of generations. So there are two different forms of a plant. There's a diploid form called the sporophyte, and then there's a haploid gametophyte phase. These are two different generations. So in animals, like us, humans, the individual is always diploid, right? And meiosis produces the gametes, which is the only time we really see haploid cells in our life cycle. But plants do it differently because they have a haploid phase, the gametophyte phase, that alternates with the sporophyte phase. And the the spores produce the gametophyte phase, and then this gametophyte can produce gametes. That's why it's called a gametophyte. The gametophyte can produce gametes, like pollen, an egg, and then that can be fertilized into a zygote, which is diploid. Right? When two haploid gametes come together, that forms a diploid zygote. So let's start from the beginning. Right? The diploid sporophyte generation of this fern produces spores by meiosis. Right? This brown arrow means that this is haploid. So we get haploid spores. That spore grows up. They don't fuse together. It's only one spore grows up into a haploid gametophyte, right, which can be larger or smaller than the sporophyte. This lasts for a very short time in certain plants. So it depends on the type of organism or the type of fungus. So the gametophyte is what produces haploid gametes by mitosis. This is still not meiosis. We're not dividing anything up. Right, we're still, a haploid organism is producing haploid gametes. Two haploid gametes have to come together to form a diploid zygote, which can then divide by mitosis to form the sporophyte. Right, the sporophyte can then do meiosis to produce spores. The spores grow up into a gametophyte. The gametophyte makes gametes by mitosis. Right. The gametes then get fertilized into a diploid zygote. I'm getting ahead of myself. The spores grew into gametophytes, which is the haploid phase. The gametophytes produce gametes that fuse together very much like sperm and egg do. And that gives us this diploid zygote. And the zygote will grow into a diploid sporophyte. And the cycle will repeat. So now let's go to male mammals. And we'll talk about sperm production. So in the human life cycle, like I just mentioned, the only time you see meiosis is to produce sex cells. Right? Otherwise, we're all diploid. All of our cells are diploid. So in human males, meiosis only occurs in the testes. And meiosis in males is part of spermatogenesis, which is the production of sperm. And this doesn't happen until males reach puberty. So this is a common diagram of a sperm cell. It's a modified cell. So a lot of things have to happen to that haploid daughter cell after meiosis for it to become a fully mature sperm. And while females produce only one egg each month, males produce millions of sperm almost daily. So meiosis is always occurring in males. So let's go through an overview of spermatogenesis. So within the testes of a male, we have special cells called spermatogonium. These are germ cells. A spermatogonium is a sex cell because it can make more of itself. It can divide and become another spermatogonium. It's like self-renewing itself. Or this spermatogonium can become a primary spermatocyte. And this primary spermatocyte is what undergoes meiosis in males. Right, the only cells in a male that undergoes meiosis is a primary spermatocyte. So again, note that this is a diploid organism, right? This is a diploid cell. At the end of meiosis, we're going to have 2 haploid cells. Then those each are called secondary spermatocytes. You can always remember primary spermatocytes do meiosis 1, right? Primary for 1. Then secondary spermatocytes do meiosis 2. And that's how we have the haploid gametes with only 1 copy of each sister chromatid. And these then become spermatids and finally sperm cells. So from 1 original spermatocyte, we have 4 sperm. 2 of these sperm will carry the X chromosome. So this male has an X and a Y. Right? So 2 of these sperm cells will end up with an X chromosome, meaning that whatever they penetrate, whatever egg they go into, right, that will make a female. Right? Because the father determines the sex in a way, right? If the dad is carrying an X, that means the baby's an X. The baby's the daughter. If the dad is carrying a Y, that means the baby will be male. The sperm does not contribute much. It contributes DNA. It contributes the centrosome to make microtubules, but that's about it. Oogenesis is egg production in females. And the ovaries contain stem cells called oogonia. Oogonium is singular. And the production of egg cells, or oocytes, is complete before birth. So you have if you're a female, you have many primary oocytes with 46 chromosomes that were formed when you were a fetus. And interestingly, these begin meiosis, but only a few continue when a female has become sexually mature. So starting from puberty, a hormone called FSH is released from the brain, and it stimulates one potential oocyte to complete meiosis. This happens roughly every 4 weeks. So let's go back for a second. A female is born with primary oocytes that actually start meiosis, but they arrest in Prophase I. After puberty, once every month, roughly, meiosis continues because a hormone is released from the brain. But again, this arrests in Metaphase II. Only if a sperm enters will meiosis and fertilization actually happen. Right, so meiosis 2 is completed only after a sperm enters. And we're going to talk about these polar bodies that get discarded. So remember I just mentioned that the egg contributes a lot more than the sperm. The egg has all the organelles, all the cytoplasm, all the enzymes, all the good stuff we need for the new zygote. The sperm is just contributing DNA and microtubules. So what we're trying to do with these oocytes is we're trying to conserve as much cytoplasm. And in the end, we're only going to have 1 mature egg instead of 4. Right? We're going to have 1 big egg with lots of cytoplasm. And we're going to end up with little cells called polar bodies that just have chromosomes in it. So we're trying to conserve, we're trying to save as much cytoplasm as possible in these oocytes. So this first division produces 2 haploid cells, but one of these cells is so much larger than the other. This polar body has the same DNA as this, but this barely has any cytoplasm and good stuff. So this will be discarded. This will die. This secondary oocyte, again, will only continue through Metaphase II and finish meiosis if a sperm enters. Right? Once a sperm enters, meiosis is continued. So Metaphase II finishes, you have Anaphase II, Telophase II, and then you have final cytokinesis, where you have another polar body discarded, and then you have your first zygote, your first diploid cell, which is the fertilized egg. And the book goes through this in detail as well. Right? So to review, we start off with a germ cell in the ovaries of a female called an oogonium. Females are XX, right? Meaning for their sex chromosomes, they only have an X chromosome. It's a stem cell, which means it can make more of itself. It can self-renew. But what it becomes after mitosis is also a primary oocyte. And the primary oocyte goes through meiosis I. And meiosis I produces a secondary oocyte with lots of cytoplasm and a polar body with less cytoplasm. This will, again, be discarded. It could even divide into two other polar bodies in meiosis II, but these polar bodies will be discarded. We don't care about these. Only the secondary oocyte will complete meiosis II if a sperm enters, and then it will do another asymmetric division where you have one large egg. A mature egg is called an ovum, and then you have a little polar body that gets discarded again. So you can look at the difference in size between the egg and the polar body. So at the end of oogenesis, we just got one ovum, only one egg cell from each primary oocyte. Compare that to male spermatogenesis. One primary spermatocyte gave us 4 sperm cells. So this is an overview from the book, right? Primary spermatocyte undergoes meiosis I. We get 2 haploid secondary spermatocytes that go through meiosis II to form 4 haploid spermatids that mature into sperm. We get 4 sperm cells from one primary spermatocyte. In oogenesis, a primary oocyte divides by meiosis, but it's asymmetric, right? In this picture, you're seeing that the polar body doesn't even pinch off from the cell, which is a possibility, right? It could be, but this is still a haploid. This is 1N and this is 1N. They're 2 separate cells. Only once a sperm enters will meiosis II continue. So now meiosis II will continue, and a secondary polar body will be pinched off. And then when those are discarded, we now have, when sperm and egg fuse, a zygote. That's diploid. Right? You have one copy of every gene from dad that came in the sperm, one copy of every gene from mom that was present in the egg. So just to bring this all together, we said that humans are 2N. We start off as a zygote that undergoes mitosis. Right? We are always diploid our whole lives. Right? This is the first cell that we are, and we always stay diploid until puberty, when we start finishing meiosis, producing egg or sperm. And then those two fuse together to form the diploid zygote. So most of the time, the zygote will just go on to form a single organism. But sometimes that zygote can split. And that would give us identical twins. So monozygotic twins are genetically identical. They're clones of each other. Because at some point, the zygote splits into 2 and gives us 2 separate embryos. And since the zygote had the same genes, that means each embryo will have the same genes. So these are clones. These are identical, and that's why we call them monozygotic. They came from one zygote. Fraternal twins have no more in common than any brother and sister, right? They have unique DNA. And that's if 2 egg cells get released for some reason, and 2 sperm cells happen to fertilize 2 separate eggs. We get 2 separate zygotes, right? Dizygotic twins are fraternal twins because you have two separate zygotes. And each zygote will then divide into its separate embryo. So, of course, they're going to be born at the same time, and they share the same uterine environment. But genetically speaking, they are just as related as any 2 brother and sister, or brother and brother, or sister and sister. So I want to now talk about how changes in chromosome number and structure can cause various syndromes in humans. So crossing over creates a lot of variation, and it's essential, right? And we need to have proper segregation of chromosomes to have the right amount of chromosomes, but so many things could go wrong in these processes. The process of crossing over and independent assortment and all these microtubules trying to connect everywhere, right? A lot of things could go wrong. And when things go wrong, syndromes can occur. The way you can diagnose or detect a syndrome is by laying out all the chromosomes of an organism, right? So in a baby, or in a fetus, I should say, it's possible to take fetal cells from the mother while she is still pregnant, and you can look at the baby's karyotype. You can stain the baby's chromosomes with a special stain, and you can count them to make sure there's the right number of chromosomes that all look like the right size, nothing's missing. There's also a lot of more high-tech options now. You can do some sequencing to figure out the structure of chromosomes, and PCR, which we'll talk about later. You can also study the pedigree of the disease. You can look at the inheritance pattern in a family to understand syndromes. But if you have the proper number of chromosomes, that's called euploidy. So hopefully when you do a karyotype, you'll see euploidy. It's the correct number of chromosomes. In humans, that would be 46. Aneuploidy means incorrect number of chromosomes, right? So if you're aneuploid, that means there is a change in the chromosome number because of an error during meiosis. And we see aneuploidy in plants and animals. And the reason why we get aneuploidy is because of non-disjunction. This is a very important term, non-disjunction. So because of non-disjunction, we get aneuploidy, right, which is the incorrect number of chromosomes in our haploid cells. So let's look at the right here first. This is a normal meiosis one. We have pairs of homologous chromosomes that separate, right, at the end of meiosis one. So this is chromosome 1 from mom, chromosome 1 from dad. Both of them, of course, got duplicated, so they have sisters. But after crossing over, they separate. We consider these haploid still, right? Because they either have the chromosomes from mom or dad. But what if they don't divide? What if the microtubules, for some reason, don't grab onto the second pair? And what if both pairs of homologous chromosomes end in one cell and the other's just empty? That's called non-disjunction in meiosis 1. So a primary non-disjunction is the actual term. So in a primary non-disjunction, that means both members of a homologous pair go into the same daughter cell. So that's really bad. What happens if, let's continue from here. This is obviously going to form just two nothing cells with no DNA at all. And then you're going to end up with this cell doing meiosis 2 and forming cells that have double the genetic information. They're going to have two of the same chromatid, either from mom or from dad. So it's just double the information that you don't want. So in a primary non-disjunction, you get 4 defunct cells. Let's look back here. Since meiosis 1 was fine, it could continue like we want, and the sister chromatids can separate, so we get 2 haploid cells. This is what we want. However, it's possible to also get a secondary non-disjunction, which occurs during meiosis 2 when the sister chromatids fail to separate, and both sister chromatids end up in one of the gametes, and one gamete ends up empty. So in a secondary non-disjunction, half of the gametes are defunct. So let's think about it. When sperm fertilizes these eggs, we're going to have different situations. We're going to have 2n plus 1, meaning you have an extra copy of every gene. This is called trisomy, right? This is an individual that will have 3 copies of a given gene instead of just 2. So that's really bad. But you can also have monosomy, which is 2n-1. Right? That occurs when an individual has only one copy of a chromosome when you should have two. So trisomy and monosomy are two examples of aneuploidy. So you should understand that, right? So monosomy and trisomy are two aneuploid states that can occur due to non-disjunction. In this case of secondary non-disjunction, you're going to end up with one that's trisomy because you had these two sister chromatids from mom plus the sperm. You're going to have one that's monosomy, that's only the sperm from dad. And then actually you're going to have two nice and healthy zygotes that are 2n, right? One copy of the gene from mom, of the chromosome from mom, one copy of the chromosome from dad. That's what we hope for. So again, non-disjunction is usually lethal, right? Because in animals, monosomies and trisomies are usually lethal. And that means that there would be either a miscarriage or you would never even know that the woman was pregnant to begin with. In general, a trisomic individual is more likely to survive than a monosomic one. So like having extra information is better than not having enough information. And we'll talk a little bit more about human conditions after the slide. I just wanted to show you another way to look at it with colored chromosomes, how the gametes should arrange and what happens in a primary non-disjunction versus a secondary non-disjunction. Right, this is what we want. We just wanted these. So trisomy 21 is when you have an extra copy of the 21st chromosome. And this is the most common autosomal trisomy seen in humans. So I'm sure we've all heard of Down syndrome before. It's recognized by certain physical characteristics, like short stature. There's an eyelid fold that's characteristic of it, a flatter face, a larger tongue. There's a large space between the first and second toes. And unfortunately, they have some heart problems and mild intellectual disability. However, they can still lead very productive, healthy lives. So this is an example of where having an extra chromosome is not that bad. There was a very endearing show on a few years ago called Born This Way that shows how Down syndrome individuals live very productive, fulfilling lives. The chances of a woman having a child with Down syndrome increases very rapidly with age. So in women that are under 30, the incidence of trisomy 21 is a lot lower than over 35. I'm going to show you some statistics that show that the older a woman is, the more likely it is for her to have a baby with Down syndrome. But it's not because of women having old age that there are people, or not because females are having babies older that there's more Down syndrome, right? Most babies with Down syndrome are born to women younger than 40, right? And actually, a recent study suggested that in 23% of the cases, the sperm actually contributed the extra chromosome, the 21st chromosome. So again, normally this is what the karyotype would look. The karyotype would not have 3 copies; it would only have 2. But in trisomy 21, you have 3 copies instead of 2, which leads to Down syndrome. You can also have a change in the sex chromosome number, meaning you get too many or too few X or Y chromosomes. Right? So non-disjunction during oogenesis or spermatogenesis can result in gametes with an abnormal amount of sex chromosomes. These are actually a lot more tolerated in humans than extra copies of autosomes, like chromosomes 1 through 22. So an example of a change in sex chromosome number is Turner syndrome, which is when females only get a single X chromosome. We refer to these as XO females. Right? There's only one X chromosome, an X. The O just indicates the absence of a second sex chromosome. This happens in about 1 in every 10,000 females. They have a couple of symptoms like short stature, delayed puberty. They're unable to have children because their ovaries and uterus are underdeveloped, so they never undergo puberty. But some were able to do IVF using donor eggs. And intellectually, they're usually normal. There might be some learning disabilities in Turner syndrome. But if they receive hormone supplements, they can lead very normal lives. So what's abnormal about each of these karyotypes? Right? It's kind of pointed out to you here, but you should be able to look. If you were a geneticist, you would look at these karyotypes and see, oh, there's a missing X chromosome. That means it's XO. This is a Turner syndrome female. Here, we have an extra X chromosome. There should only be an XY. So we call this is called Klinefelter syndrome, when you have XXY, right? This happens actually a lot more commonly than Turner syndrome. This can happen in 1 to every 500 males. XXY is Klinefelter syndrome. So this is, right, so it's between 1 to 500 and 1 to 1,000. So we'll say it's 1 to 750 males, approximately. Compare that to Turner syndrome. And then Klinefelter, so you could also have triplo X, which is very tall stature females. So the presence of a Y chromosome is what makes a human male. So if you have no Y chromosomes, you're considered female. There's also XYY, which is considered a male because you have Y chromosome. That's called Jacob syndrome. And that's a little more common than Turner syndrome. So here's just some examples of sex chromosome abnormalities. I would recommend knowing at least an example of one. And the book goes into a lot more details of that. Okay, so how do errors occur within chromosomes? We just said that aneuploidy is when you have an incorrect number of chromosomes that occurs by non-disjunction. But what about a change within a chromosome? Right? So a mutation within a chromosome can have major effects on the organism as well. Suppose we want to have a chromosome with the following genes, A, B, C, D, E, F, G. We can have a deletion, which just completely cuts off certain genes. Right? That would not be good. That usually happens during crossing over sometimes. Or if there's a break in the chromosome, it can mean that a whole segment just kind of flies away. And a deletion will almost always cause abnormalities because you're missing essential genes. You can also make copies of genes. So here it says DEF, DEF. So in duplication is when you have the presence of a chromosomal segment more than once in the same chromosome. You may not cause physical abnormalities depending on which genes are duplicated. It's also possible that you can change the sequence. You can flip around the sequence of the genes within the same chromosome. So it says A, B, C, F, E, D, G. Right? So this segment of a chromosome has been turned around 180 degrees. But this doesn't usually cause many problems in inversion. So usually there are no abnormalities with this. You can also have what's called a translocation, in which two separate chromosomes switch segments. So this is like crossing over between non-homologous chromosomes. We do not want that to happen, right? So when one chromosome segment moves to another chromosome segment, that's called translocation. The translocation can either be balanced or imbalanced. So if there's an equal swap of genes, that would be a balanced translocation. This is not an equal balance of genes. This would be called an unbalanced translocation, right? So you have extra genetic information on some and not enough genetic information on others. So translocation is pretty bad. So these are just other ways to look at deletion, duplication, inversion, translocation. You should know all of those terms. An example of a deletion syndrome in humans is Williams syndrome. And that happens when chromosome 7 loses a tiny end piece. So a tiny end piece of chromosome 7 is lost. That contains the gene for elastin, which is a protein that has many effects, or many roles in the body. So with elastin missing, there are effects of the cardiovascular system. Also, the skin ages prematurely. There are mild to moderate intellectual disabilities and some distinctive facial features. So that's Williams syndrome. An example of a translocation syndrome is Alagille syndrome. And that results when chromosomes 2 and chromosomes 20 switch genes. So they actually switch genes. So Alagille syndrome is not fatal, but the deletion from chromosome 20 can cause a lot of abnormalities of the eyes and internal organs. Specifically, the internal organs like the liver and the heart can be affected. And there are this clubbed fingers is a result of Alagille syndrome. That's a way to see Alagille syndrome. The translocations can also cause certain kinds of cancer. So translocations are usually bad. So this is a table that shows all of the different human live births that have extra chromosomes. So approximately 1 out of every 155 children have some kind of major chromosomal abnormality. The most common is Down syndrome. So 13 out of every 10,000 live births have this mutation, or this extra 21st chromosome. Right, so that means I have a total of 47 chromosomes. So it's a trisomy, right? 2n plus 1, right? And remember, n equals 23 in humans. You could also have trisomy 18 or trisomy 13, which happens a lot more rarely. So you have only 2 out of every 10,000 individuals being born with 3 copies of the 18th chromosome or 3 copies of the 13th chromosome. Then you have about 5 cases each out of 10,000 for XXY, which is Klinefelters in males, XYY, which is Jacobs in males, or XXX, which is called triple X syndrome in females. And then it's very, very rare to get XO, Turner syndrome, 1 out of every 10,000. It's very rare to have Turner syndrome. And again, this is another way to look at that information based on trisomies, right? So sex chromosomes, you can either have 1 X chromosome. This is XXY, XYY, XXX. So you can see that translocations over here, again, a lot of them, there are not many fetuses are born that can actually survive having a translocation. So out of 100,000 fetuses, 8,000 had chromosomal abnormalities. And out of those 8,000, only 500 were born alive. Right? So about 500 out of 8,000 were able to survive with the chromosomal abnormality, but 7,500 underwent spontaneous abortion. I'm not going to go through this example in this lecture, but it's interesting if you're curious that sex occurs in worms as well. And worms can choose between either asexual and sexual reproduction. So what they did was they exposed these worms to either evolving bacteria that always change, or a fixed bacterial strain that doesn't change. So what do you think? Would a worm exposed to changing bacteria be more interested in sexual reproduction or asexual reproduction? Right? Compared to dead bacteria. So you can look at this, you can pause here and look at this graph more closely, but what they saw is that when you expose parasitic worms to bacteria that are always evolving, the worms choose to have sex. So bacteria induces sexual activity in worms. And then again, why are worms having sex when there's bacteria around? Well, let's think about it. The worms are trying to maximize their chance of survival in case the bacteria are infectious or dangerous. So by mixing and matching genes in meiosis, maybe one of their children, baby worms, can have an advantage that can survive in the ever-evolving bacteria. So this is why sex is advantageous, right? Worms that can only reproduce asexually were not even able to survive. So worms that only reproduce asexually all died, actually. So sex is part of life. Sex is a requirement of life, right? Sexual reproduction and meiosis can increase the evolutionary fitness of not only worms, but of all species. So that is the end of chapter 10. Please complete the lecture review assignments, and the next quiz will cover chapters 9 and 10. So mitosis, cancer, and meiosis.